Experts unlock the key to photosynthesis, a find that could help us meet food security demands. The study was conducted by Lorna Malone and the rest of her team at the University of Sheffield. Presentation was done by me, Alex Hansen. So the background. With the constant threat of global warming looming, as well as a rise in pest resistance to insecticides, and other pesticides, and an ever-growing world population, food scarcity has quickly become one of the more important issues in today's world. Scientists have been trying to produce more crop yields of bigger crops in hopes of combating this problem, but haven't had the best luck yet. Cytochrome B6F. Cytochrome B6F, abbreviated CYT-B6F, is a key complex in photosynthesis. Located between photosystem 1 and 2, it's responsible for linking the electron transfer between photosystems, as well as creating a proton gradient for ATP synthesis. This makes cytochrome B6F a key molecule in the production of ATP. A new study of cytochrome B6F in spinach found another function of the complex, its ability to alter a plant's photosynthetic efficiency. This means a plant can adjust its internal processes in response to potentially harmful environmental factors. It also means that they can live in harsher conditions. So the study that was conducted. Lorna Malone and her team's research provided necessary insight into the cytochrome B6F complex, which will lead to new developments in preventing the food scarcity problems we now face. Not only did the team find new data into the role of cytochrome B6F as a proton battery, they also found an additional role. By utilizing a single-particle cryo-electron microscopy of a spinach plant to create a high-resolution structural model, they determined that cytochrome B6F functions as a sensor of environmental conditions, like that was mentioned in the previous slide. So here we can see the model that was constructed by Lorna Malone and her team. We are going to be focusing on structures A through B and C. So model A is a top-down view of the cytochrome B6F complex in spinach, found using the single-particle cryo-electron microscopy we talked about in the previous slide. And this illustrates what Lorna Malone's team was looking at when they came to their conclusion. The green parts in the image, located right there and right there, are the cytochrome B6F complex, which is where we will be focusing. As you can see, it's located in the stroma, which captures light that cytochrome B6F then uses in the proton gradient. Figure B is the same molecule, simply shown from the luminal side, and figure C is the same molecule but shown from the stromal side. Figures D, E, and F are simply drawn model representations of model A. So the accuracy of the findings. In class, we learned about the proton gradient forms between photosystems 1 and 2, and so reading this article and learning about cytochrome B6F makes sense. Furthermore, this article was published in Nature.com as well as Science Daily and on the University of Sheffield's website, meaning the validity of these results is accurate. This presentation was made by Alex Hansen within the context of Bio 282 Conceptual Approach for Biology Majors 2, ASU 2nd semester 2021. Thank you for your time.